Good afternoon to everyone present here. I'm so privileged and grateful to have been presenting a paper in the 5th Annual Congress on Plant Science and Biosecurity. So today I'm going to present a paper entitled Evaluation of Genetic Variability and Trait Correlations Among Landraces and Traditional Varieties of Small Millets from the Hilly Regions of Tamil Nadu. So I am Dr. J. Lydia Pramita. I'm currently working as an assistant professor in the School of Agricultural Sciences, Karunya Institute of Technology and Sciences, Coimbatore. So coming to the background of the study regarding the importance of the study, saying to the Indian food industries, we have three major importances. First, related to the cultural richness. So each state in India has a specific cuisine, specific crops which are native, and each of them have their own potential. And nowadays, since the market is expanding, we can see that as far as now, we are just dependent on three major crops like rice, maize, and wheat, whereas there are around 2 to 3 lakh edible plant species. So this gives us a wide opportunity to have a diverse diet and a diverse cultivation. The third one is evolving consumer preferences. So now, other than rice, maize, and wheat, the consumers are moving towards diverse crops. So it is now a favorable situation to bring back the forgotten crops, which were once leading in cultivation as well as in nutrition. Coming to the history of the small millets or the food choices and its relation to the green revolution, we can see that before green revolution that has happened, the major crops that were cultivated in India were rice, sorghum, pearl millet, and all of these small millets. Mostly a major preference was given towards the traditional rice varieties as well as the millets. So it is known that there were around 1 lakh rice land races prior to the Green Revolution. So the major features prior to the revolution were the farmers had a very smaller plots, organic husbandry, fallow fields were left out, there was lower use of fertilizers and imports, and frequent famine was there, and it was mostly the agriculture that was practiced before Green Revolution was driven by the energy which was provided by the humans and animals. Whereas coming to after Green Revolution, we could see that there is a dramatic increase in the cultivation of rice, wheat, maize as well as pulse, and due to which there was a decline in the cultivation of coarse cereals as well as the millets. There was higher utilization of fertilizers and pesticides, and more preference was given towards the yield, due to which there was a dominance with semi-dwarf varieties of rice as well as wheat from irri as well as millet. So due to this, what happened is the traditional cultivars were actually replaced with the modern cultivars which had a narrow base for yield and its attributing traits. So if you see before Green Revolution, there was a low productivity in crop, monocropping was followed, traditional varieties and land races were more preferred, and traditional practices were followed. The cultivation practices were solely dependent on rainfall, and a conventional system of irrigation was used. Traditional practices, local sales were used. Millets and traditional rice were the most cultivated across countries as well as in India. There was no importance given to high yielding varieties or stress tolerant one, even though there was famine prior to Green Revolution. Now coming to after the situation of Green Revolution, a double cropping or monocropping was followed. Farmers started to practice a lot of cropping systems. Higher productivity of crops was there due to the preference of high yielding varieties. Improved farm implements, market development was there. The food policy was there, due to which we had a triple production of grains in case of the major staples, that is rice, maize, as well as wheat. And millets were consumed, which were the major key players prior to Green Revolution. The cultivation of millets after Green Revolution tended to decrease. So this is where we now come. The current situation is that we are trying to bring back the forgotten crops or revival of the forgotten crops, especially the millets pertaining to Tamil Nadu. So this study was conducted with the traditional land races and varieties which were collected from the hilly tribes of Tamil Nadu. The main objective of this base study is that we are collecting the underexplored or underexploited small millets from the hilly tribes, and we are going to evaluate their morphological and yield attributing traits. We are going to also observe their nutritional parameters, and we are going to perform a diversity analysis and group them into high yielding, low yielding as well as average yielding traits. We are also going to explore their cooking quality, how far they are amenable for cooking compared to the recently released varieties. So these are the three major objectives of the study. So coming to the materials that were used for the study, there were around 30 land races as well as varieties which were used, in which there were 17 little millet types which were collected from different hilly regions, western as well as the eastern Ghats of Tamil Nadu. We collected the land races. So we had the land races and varieties of little millet. We had the land races and varieties of finger millet, foxtail millet, and one landrace of proso millet as well as barnyard millet was also used. So seeing this is an overall schematic map of the regions in Tamil Nadu from where these varieties were cultivated, and this is a schematic presentation of the experimental view plot which was raised in Karunya Institute of Technology and Sciences. These were the set of traits which were recorded among the 30 germplasm lines which were raised in a randomized block design in 3 replications. So there were 10 morphological or quantitative traits which were recorded, and there were 12 qualitative traits which were identified based on the dust descriptors for the specific millet crop. And finally, the identified or the selected land races and varieties based on their yield and cooking quality. 5 nutritional traits were recorded. They were food protein, total flavonoids, iron, zinc, and magnesium. So also in addition to this, as per our objective, we also have seen the cooking quality as well as the physical parameters. So weight of 1000 grains, bulk density, the volume, and weight of soaked grains, volume of the soaked grains, bulk density, hydration capacity, swelling capacity. And pertaining to cooking quality, we had cooking time, cooked weight, volume of the cooked grains, hydration capacity of the cooked grains, as well as the popping yield. So this picture displays the differential architecture, panicle architecture of the released varieties as well as of the land races. We could see that Co4 Samai is a variety released from Tamil Nadu Agriculture University for little millet, and the rest of the genotypes represent the land races. Chittan Samai, Pakulam Karinjamai, Kolundan Samai, Kottu Samai, Kottara Samai, and ATL1. So these are the different cooking quality parameters which were were analyzed in the lab, and the above we could see the rest of the little millet land races and their panicle architecture. Although there were not much variations for the qualitative characters, there was a significant variation among these small millet land races, especially for grain color as well as thousand seed weight. So this picture could depict the variation in the grain color. So we had different gradations in brown, yellow, light brown as well as gray color of variation in grain color was seen. And this is the overall image of the cooking quality. So all the small millet germplasm was subjected to the cooking, and the time duration and the volume of expansion, how far they are getting cooked in a limited amount of time was analyzed. So coming to the results, so the 30 germplasm lines were subjected to a variability analysis to see how far their phenotypic, genotypic and environmental variability is there. And we could see that among the 10 quantitative or the morphological traits, the highest phenotypic coefficient of variation was observed in days to flowering. So we could see that days to flowering had the highest PCV and GCV, and followed by that we could also see that a higher PCV cum GCV was there in grain yield per plant. The number of basal tillers also had a higher PCV and GCV. When we see all the set of traits, one thing we could note that The environmental coefficient of variation was almost low, that is less than 10%, which depicts that there was a minimum environmental influence. Thus, there is an effective genetic variability which could be amenable for selection in further breeding programs. So when we speak of selection, and it would not be possible without heritability and genetic advance as percent of gene. As we all know, heritability is the percentage of transmissibility of a trait from one generation to the other, and genetic advance presents the effect of selection gain that happens after each round of our selection. So we could see that both heritability as well as the genetic advance over mean was higher for all the traits. Thus, there can be an effective selection that can be done for yield attributing traits from this set of land races, and they can be further proposed for yield attributing breeding trials.
Speaking to the correlation analysis, we could see that the grain yield per plant was significantly and positively correlated with all the morphological traits except for days to flowering and peduncle length. We could see that the highest significant correlation was observed with grain yield per plant as well as number of branches per panicle and 1000 seed weight. Except for days to flowering and peduncle length, all the traits can be considered as the yield attributing traits. Since the grain yield plant was negatively correlated with days to flowering, and it suggests that when we breed for early flowering types there will be a compromisation in the yield. So while going for breeding for early flowering types in this germplasm, we have to be very careful on seeing the yield attributing traits. Coming to the flag leaf length and flag leaf width, we could see that it was observed to have a positive correlation with all the traits, although the values were not significant. The flag leaf width was positively correlated with flag leaf length, and 1000 seed weight also we could see that it had a similar variation as that of the grain yield per plant except for days to flowering and peduncle length. The rest of the traits had a positive correlation except for the plant height. So thus this exhibits that the basal Taylor's plant height, flag leaf length, flag leaf width, peduncle length, number of branches per panicle and 1000 seed weight may be considered as the yield attributing traits when considering this germplasm.
So coming to the next analysis, we have the diversity analysis where we group the genotypes based on the Mahalanobis d-square distance. This is the diversity clustering pattern for the quantitative traits. So here we can clearly see that the released varieties, that is CO4 Samai and ATL1, which were released from Tamil Nadu Agriculture University. So this actually clustered or grouped, this was grouped as a separate cluster in the diversity analysis. And the next cluster we could see that the cluster 2, it had all the land races which had a higher grain yield as well as a higher 1000 grain weight. So we could see that bolded grain types as well as a high yielding ones all grouped together in the second cluster. We had a third cluster and the fourth cluster. Individually it was Kalman Samai, and the rest of the genotypes came under the fourth and fifth cluster. So this is the clustering pattern which happened in the morphological traits for the D-square analysis. Coming to the qualitative traits, as I said earlier, there was not much variation other than the grain color as well as the grain weight. So we could see that there were minimum amount of clusters in which we could see based on the inflorescence shape, the genotypes were grouped into 3 clusters. So this clustering has happened based on the contribution of the different traits towards their clustering pattern. So coming to the quantitative traits, that is the morphological traits, we could see that the major contribution was given by the 1000 grain weight, and followed by the days to flowering was a major key role in grouping the genotypes into different clusters. Whereas coming to the qualitative traits, it was the culm branching as well as the inflorescence shape played a major role in grouping the genotypes into different clusters. So based on their mean performance, the high yielding genotypes were ATL1, Chittan Samai, Periya Samai, Pakulam Karunjamai and Vellai Samai. So this ATL1 summary, these 5 genotypes were actually subjected to nutritional analysis, and we could see that ATL1 summary compared to the rest of the land races was superior to the nutritional parameters. It almost had higher crude protein, flavonoids, iron, zinc as well as magnesium. Whereas coming to speaking of the land races, we could see that the highest crude protein was present in Paculum caringiamae. So this genotype was also noted to have a higher fodder yield. Among the flavonoids, we could see that again we had the Paculum caringiamae as the highest one. And considering the iron, so apart from the higher values that were given by ATL1 Samai, we had Periasamai, which was also superior in iron, and zinc was higher in Periasamai again, and magnesium was also higher in Paculum caringiamae. So these were the nutritional significance or the nutritional analysis of the identified land races. So these land races were found to be higher yielding, and when subjected to nutritional analysis also, they had a higher crude protein, flavonoids, iron, zinc and magnesium. So concluding to the study, we could see that based on their mean performance as well as their clustering pattern, the ATL1 Samai and Co4 Samai, which were the released varieties, had a higher yield. So after that, following them, we had Perinjame and Chitanjame among the land races of little millet, which had the highest yield. Speaking of early flowering, most of the traditional varieties or land races, they are regularly late flowering. But among the set of germplasm, we were able to see that Javad local Samai, Kottaru Samai, Kolundana Samai, Chitan Samai, IR20 Samai, Co4 Samai, Siru Samai, IR8 Samai and Perin Samai were observed to be early flowering types. The rest of the genotypes were late flowering. And for bolder types, that is a higher 1000 grain weight, the genotypes namely ATL1, Chitanzamai, Kottarasamai, Kofor Samai, Peria Samai, Kalman Samai, and Javad local Samai and Perinjamai were recorded to have a higher bold seeder. So they can be used for developing bold seeder types in small millets. Next, coming for the performance of yield, overall performance of all the yield attributing traits, we could see that Chitanzame and Perinzame had a higher performance for yield attributing characters. Regarding the qualitative traits, although there was no variation for among all the genotypes, the highest variable qualitative traits were culm branching. There was a variation observed for branching type, inflorescence shape, lodging as well as shattering in seeds. So there were both non-lodging and lodging types. There were both non-shattering and shattering types. The high yielding genotypes were almost obviously the released varieties, recently released varieties, that is ATL1 and Co4 Samai. Both of them had a diffused inflorescence shape with an open intermediate panicle structure. So we could say that this diffused inflorescence shape and this open intermediate panicle is a key panicle architecture for developing high yielding varieties in small millets. So based on the mean performance other than the little millet, so we had the Co15 Ragi, Co7 Tinai, that is foxtail millet, Kotatara Panivaragu, they had a higher yield. And among the rest of the 13 small millet genotypes, grain color and grain shape were found to be varying. And regarding the cooking quality, for hydration and swelling capacity, which are the key players for a cooking, a good desirable cooking quality, we had a set of genotypes, that is Kolundana Samai, IR20 Samai, Kottu Samai, Pakulam Karinjamai, Perungulam Samai, Perum Kelvaragu, Kottathara Tinai, Kottathara Kudiraivalli. They had a higher desirable cooking quality parameters. So in a limited amount of time, it was found to have a higher hydration and a swelling capacity, which is favorable for cooking. So overall, these are the genotypes which can be further processed or further recommended or popularized among the public for cooking purposes. Regarding the popping yield, nowadays the small millet pops are ruling the market. So when we tried out regarding the popping percentage, we would be able to see that Periyasamai and Perunkelvaragu had a higher mean performance for the popping yield. So they can be further promoted for developing popped products in small millets. So regarding the genetics advance and heritability, all the traits that were recorded had a high value. So we could say that there is a predominant role of additive gene matching and selection can be done effectively and it will be rewarding. So among the five clusters that we have got in grouping the genotypes, the maximum intra-cluster distance was recorded in cluster 4. So we could observe that other than the person contributing traits, the genotypes that were grouped in cluster 4, for the rest of the traits, it was highly variable. That is why the intra-cluster distance was higher. And regarding the intercluster distance, the maximum intercluster distance was found between cluster 2 and cluster 5. So the genotypes in cluster 2 and cluster 5, they were Kolundana Samai, Ayareta Samai, Siru Samai, Perungulai Samai and Kalman Samai. They can be used as parents in hybridization programs to have a superior recombinance. So considering the percent contribution of traits, we had 1000 grain weight, days to 50% flowering, grain yield per panicle had the major contribution towards the clustering pattern. So these are the findings of my study and I would like to conclude my presentation with an acknowledgement, sincere acknowledgement and gratefulness to Karunya Institute of Technology and our team, genetics and plant breeding team, both the PG students as well as my colleagues who were with us in successfully helping us in carrying out the collection of these germplasm from the hilly tribes. So with this, I sign off. Have a nice day. Thank you for giving me this chance.